First, I want to pose the scenario and remind that immune processes are schematically divided into two sequential steps. The first one, which is the first line defense, which is innate immunity, which is non-specific, rapid, and concern every signal of danger. And if such process is not resolved, so we have the second part, which is the adaptive and specific immune responses. And all the molecules which are produced during both the two processes are under genetic control. This is what we call the immunogenetics. And immunogenetics, the analysis of immunogenetics can allow to identify susceptibility status to or protection against various diseases, including psychiatric disorder and bipolar disorder, which is a frequent disease, chronic and progressive, multisystemic, as you will see because of the presence of a lot of comorbidities, which is highly heterogeneous and often accompanied by a chronic inflammatory background. The progression of bipolar disorder is characterized by a progression towards low cognitive status and accumulation of medical comorbidities like diabetes or thyroid disorder. And we have to notice that immune dysfunction are observed during all the progression of the disease. Such immune dysfunction are reflected and exemplified, among others, by inflammatory marker, either at central or at peripheral level, by a high prevalence of excuse me, autoimmune disorder, and by the additive effect of both infection and autoimmune disorder in the incidence of the disease. So, the question we pose, what could be the origin of such immune dysfunction? It is evident if we consider that the high irritability of the vast majority of psychiatric disorder and of also of bipolar disorder, and if you consider also the part of the environment in the development of the disease, all these features means that probably complex interaction between gene and environment at work. Hence, we can consider bipolar disorder as a multiple hit model, starting during the early or prenatal period, and after a series of stressor events, the disease appear and progress. This slide summarizes what you can imagine in case of absence of the genetic defect. You can see that in case of such absence of genetic defect, there is a resilience because all stressor, all the infectious event will be resolved. In case of genetic defects in the immune response mechanism, then we can pose the question if there are differences in the immunogenetic explaining the inter-individual susceptibility of patients. If you consider environmental factor, the best known for the moment is the infectious events. And this infection concerns all types of infection, including viruses, bacteria, or parasites. And they occur during perinatal period or even later in life as showed by lifetime infection. The best example is the Toxoplasma gondii, when there is a series of studies showing without any doubt the implication of this parasite in the risk to develop bipolar disorder. Hence, how can we explain the persistence of infectious stigma, and how can we explain the pathological role of infection later in the development of bipolar disorder? We use the strategy called evidence-based belonging to both arms of immune responses, focusing on functional relevant SNPs and SNPs with proven association with common disease. We did a scan of the potent player of the immune responses during both phases, and we focused on pattern recognition receptor during the innate immune response, on the modulation of overall immune responses, and then on the adaptive immune responses by studying the polymorphism of the HLA cluster. We start by the PRR, which are a logical source of candidate gene for bipolar disorder because they are frontline for protection against pathogen. They are also frontline for protection against endogenous molecule, and as they sense what we call DAMP, damage-associated molecular pattern, and are represented in all systems and all cells of the body, including brain. The first one we studied, which is the best known and the most studied, is TLR4, and it's specific for LPS recognition for bacteria, but can also recognize a various number of other pathogens, including fungal, viruses, and parasites. We identified among 6 SNPs studied that 2 of them at homozygous status were associated with bipolar disorder, especially with the early onset form of bipolar disorder. The 2 SNPs were previously associated with transcriptional regulation and are predicted to influence mRNA binding site. We found also 2 SNPs associated with thyroid disorder. So, within the same gene, we can unmask association with inefficient anti-infectious response and with autoimmunity, the yin and the yang of immune dysfunction. And we have hypothesized that the 2 first associated SNPs were associated with low expressor genotype of TLR4. So this low expression may render susceptible the patient to infectious event. In the second example, we know that the 2 SNPs change the conformation of the TLR4 molecule, and in this case, we can fall in the theory that after stimulation of an antigen and repeated stimulation, there is a breakdown of tolerance and appearance of autoimmunity. The second locus we analyze is TLR2, which is also extremely important because he sends every kind of pathogen receptor, of pathogen molecular pattern, and he works also as homodimer or heterodimer with other TLR, so covering a vast repertoire of signal of danger. And here again, we identified a SNP variation at homozygous state associated with early onset bipolar disorder. And this SNP is associated with low production of cytokine, so we hypothesized that here again, inefficient anti-infectious response may be at work. So, after reviewing the sensor, we posed the question about, because it is evident that inefficient anti-infectious response will lead to inflammatory process and to a pro-inflammatory status. So we posed the question, what about immune modulation? So we analyze one of the most important molecules in such setting, which is HLA-G, which is known to be immunosuppressive and to have tolerogenic properties. And here again, we identified strong association with a high production of the immunotoleric or immunotolerogen molecule in bipolar disorder patients. So, if we see overall our results on both innate immune process and on the modulation, we can hypothesize that when there is a diminution of inability to mount efficient response, there is augmentation of the risk to develop bipolar disorder. The second type of environmental risk factor we analyzed is the early and severe stress. It is well admitted that severe and early stress during childhood is related is further related with an increase of a lot of metabolic disorder and a lot of disorder, including psychiatric disorder. Because I don't have the slide here, but stress is mediated also by pro-inflammatory process. So first, in our group, we demonstrated that childhood trauma is associated with bipolar disorder. Then, we know also that childhood trauma is associated with early onset bipolar disorder. So the question we pose is there any relationship between what we observed in terms of immunogenetics of TLR2 and TLR4, because both were associated with early onset of disease, and with trauma during childhood period. And we analyzed the combination, the possible additive effect between these two risk factors. And we found indeed that in individual having the genotype at risk for TLR2 at homozygous state and having a history of sexual abuse, this individual develop the earlier onset form of the disease, as you can see. I don't know if it works. Comment ça marche là? So, just to summarize the situation after our thinking, our finding, and after our hypothesis, bipolar disorder can be considered as a series of stress during life. If this stress appears in immunogenetic susceptible background, this will lead to inflammatory process, and this inflammatory process further may promote autoimmune disorder. At this stage, the question also is what could be the link, the functional link between innate and adaptive immunities and potentially explaining pro-inflammatory process amplification. For an immunologist and for histocompatibilist, the answer is clear. It's the HLA system, which is the system at the crossroad of infection, inflammation, and autoimmunity. And the sequential possible event, the sequence of the event could be infection, non-resolved infection, inflammation, and after that, autoimmunity. HLA system is hosted by the CMH, the complex, by the MHC, major histocompatibility complex in the chromosome 6. It is considered as the most polymorphic cluster in the human genome, with more than 70,000 alleles, and is the most associated cluster with common disorder. The cluster is divided into three classes: the class 1 and class 2 classes, which are the classes of antigen presentation, which are the speaker to the lymphocyte, to T lymphocyte, and the class 3 region, which encompasses the complement region and which is involved in inflammation and also in brain development. This is one of the reasons for what we focus on the HLA system, because HLA system, I represented only the top hit of association in human disease, and you can see that at this time, this slide is about, I think we did this slide 10 years ago, and at this time only schizophrenia was identified as mediated by, linked to the polymorphism of HLA. This is also another reason to focus on HLA system, as series of GWAS clearly identified the association between the MSC region, which hosts the HLA region, and all the major psychiatric disorder, including BD. Due to the high polymorphism of the system, dealing with 7,000 allele is impossible, because you need more than 10 million individuals to put in your study. So we take advantage of selective pressure of the selective constraint, making that the MSC is transmitted in block and transmitted from several ancestral haplotypes. So we did the analysis on very homogeneous patients, at least from three generations of French descent, which is extremely important in terms of analysis in HLA and associations story. And we focus first on the overall risk, and second on phenotype, clinical phenotype, evoking inflammatory process. As I told you, we choose to analyze not the diversity of each locus or for each allele, but the diversity of the whole of or subpart of the HLA cluster haplotype. So, in terms of overall risk, we observed that given HLA-derived haplotype derived from HLA-44 is associated with protection against bipolar disorder. This haplotype belongs to a family of haplotype known to because ancestral haplotype represent the function of the whole cluster, and for many ancestral haplotype, we know what is the significant weight and the significant meaning. So this haplotype was related with diminished TNF activity and with diminished response to AIDS. So our protective status here could be explained by a decrease of inflammatory process. Then we focus on rapid cycling, and we identified a full haplotype, which is the 57-1 haplotype, very well known haplotype in all the settings, as it is important in development of multiple sclerosis. It is considered as a pro-inflammatory haplotype, and in one of the best associated allele, which is B57 allele, is associated with strong adverse reaction in patients treated by retroviral drug, antiretroviral drug. Second finding concerning rapid cycling concern a very well-known haplotype for immunologists, which is called the Autoimmune Haplotype, the HLA 8.1. This haplotype is the most frequent in Caucasian, is the most associated with autoimmune disorder, so it is defined by a natural inflammatory status even in healthy individual. And in our observation, we could relate such susceptible status with the pro-inflammatory properties of this haplotype. Then We also observe association with suicidal behavior or history of suicide, and we found the same autoimmune pro-inflammatory haplotype associated with suicide. And this also could be explained by a lot of literature showing that there is a real intertwining between inflammation and suicide. Finally, we also observed association of HLA diversity at haplotype level and mood polarity and psychotic symptom at onset. We identify two almost identical haplotype derived from the HLA 7.1 and associated with manic episode at onset and associated with psychosis episode at onset. If we look for the previous association of this haplotype, we found that this haplotype was associated with slow progression of AIDS, means pro-inflammation. The HLA DRB115 allele, I regret that MO is not here. 15 is the best associated with multiple sclerosis, which is a really intertwined disease with bipolar disorder. And our team also reported many years ago that such haplotype is protective against severe bacterial infection. The most interesting finding, it's a very recent and very last survey identifying this haplotype as a strong risk factor for Alzheimer's disease and an inflammatory neurodegenerative disease. And in the paper also, the author discuss the overlap with multiple sclerosis. So here again, pro-inflammatory processes are evidently at work. We can, for the conclusion, what we can conclude, what we can not conclude but hypothesize. The first pass of innate immune response exhibit defect in certain condition in individual otherwise predisposed at psychiatric level. In case of this defect, then they opened the scene for the work for the amplification of the pro-inflammatory status by the adaptive system and by the HLA system. And this will allow finally to the appearance of the autoimmune disorder or metabolic disorder and all what we observe in terms of somatic problem. So, just to finish light, all of these steps could be targeted by simple and already used drugs, anti-inflammatory drugs, by repurposing such type of drugs to at least alleviate or improve the somatic problem observed in the disease. Thank you for your attention.